This tutorial will explain how to upload, organize, annotate, and analyze disk diffusion assays and e-tests using J-AST. Begin by starting the desktop application or logging into the web app. Create a new project by clicking the New Project button. Enter a name and ensure that the project type is set to Timeline. Please note that later on you can change the name and project type at any time. After the project has been created, open it by clicking the corresponding entry. To add images, open the Upload panel by clicking the Upload button located at the top left of the application. Click the small plus button and stage images for uploading. Then click the Upload Now button to start the operation. Click the Upload button again to close the panel. At this point, the images are organized into a dedicated drawer as J-AST does not know yet how to organize them into a timeline. For this, you will need to attach metadata to the images. First, select all unsorted images by clicking the provided button. Then, switch the mode to the Process tab. J-AST can automatically extract metadata from the image file name through rules. Run the Autofill Metadata tool on the images, which will open a configuration dialog where you can set up how J-AST retrieves the metadata. The tool will split the file name into parts and use them to fill the metadata fields, specifically the assay type, experiment, sample, and time point. For the example data we provided, the correct configuration is as following. The assay type is dynamically extracted from the third part of the file name. The experiment identifier is stored within the second segment, while the sample is extracted from the first. The time point is in the fourth part. The editor also allows fixed metadata fields. For example, if you know that all images are e-tests and no such information is located within the file name. If you have differing file name layouts, you can also just run the tool on a subset. Also, you can at any point just edit the metadata manually through the Edit tab. The metadata now should be shown as colored badges next to the image. With all metadata set up, you can let the software organize the images automatically into a timeline for you. To do this, select the Auto Sort by Metadata operation in the Process tab. Alternatively, you can also sort images manually by dragging them around. As this becomes cumbersome for larger datasets, and it is good practice to keep track of metadata, we highly recommend to maximize the use of automated functions. The operation requires a single input, namely the correct time order. For this dataset, images are either from a 24-hour or a 48-hour time point. Add them into the list and ensure that the order is correct. The data now should be properly organized for all the following operations. If you made a mistake with the sorting, you can use the Move to Unsorted tool to move all selected images back into the unsorted category. Before starting with the analysis, images need to be further annotated with specific regions and additional metadata. Let's begin with a mask that highlights the plate area. For the provided data, the automated operation works well. To run the fully automated operation, go to the Process tab, open the Plate category, followed by selecting the Auto Detect Plate operation. By default, the workflow will automatically calibrate the physical pixel size. For that to work as expected, ensure that the plate diameter is correct. Please note that the speed of image analysis workflows depends on the number of images, algorithm complexity, and compute capabilities. After the operation has finished, review the red annotations shown on the images. These should outline the plate. The pixel size calibration can also be executed as dedicated operation if you provided a manual plate annotation. Many operations that work with disk diffusion assays require information about the disk location. Due to the high variation in image properties, we offer 3 algorithm variants. For our test dataset, the version 1 algorithm is sufficient. Ensure that the disk is correctly outlined with a green marker. To detect the E-test strips, you can use the Auto Detect E-test Strip version 1 algorithm. This operation looks for a black or white outline that surrounds the strip's content and assumes a certain aspect ratio. Our test data has white outlines. We leave the aspect ratio as the default value of 11. Unfortunately, E-test detection is non-trivial and highly sensitive to image contrast, noise, and artifacts. This is where the interactive tools will cover the gaps left by automated operations. Select an image that has a wrong strip annotation and go to the Edit tab. Then scroll down to the field for the annotation and click the Edit button. As the annotation in the shown image is unrecoverable, use the Clear tool to reset the mask. For E-test strips, the Polygon tool is perfectly suited. Enable it and click the 4 corners of the strip where at the last corner a double click is used to confirm the outline. Do not forget to click the Save Annotation button to finalize the edit. The outline now should be updated. The Disk Image R algorithm also requires the shape of the zone of inhibition. Let's draw it manually by clicking the Edit button located in the ZOI Shape panel. Use the Pen tool with a large width to roughly outline the ZOI. Focus on the outline itself. Symmetry and filling will be handled in the next steps. Then use the Fill tool to fill out the shape. To make the ZOI symmetric, switch to the Symmetry tool. Draw the symmetry axis through the center of the strip and observe how the tool perfectly symmetrizes the shape. Again, use the Save Annotation button to finalize the edit. Continue by annotating all remaining images with the exception of the ZOI shape for the second time point. Here we will use instead an operation that automatically copies over the ZOI shape from earlier to later time points, automatically correcting for differences in rotation and placement. With all images being annotated, we can finally apply the Disk Image R analysis. Select all images to analyze and navigate to the Process tab. Within the Analyze category, you can find the Disk Image R analysis operations for timelines and single images. Choose the timeline variant and keep the default settings. Wait until the workflow finishes. Then click the Results button at the top right to open the list of all results. Select the Disk Image R result entry. Here you can download and browse through the results. For example, you can open the all-in-one statistics file to view it directly from within the application. Finally, let's use the automated workflow for finding the minimum inhibitory concentration. For this, each E-test additionally requires information about the layout of the strip. As all strips in the test dataset have the same layout, we can begin by selecting all the E-test images after clearing the current selection. Click the Set E-test Strip Preset button, select the E256 option, and click OK. You should now see a red metadata badge on all E-test images, confirming that the preset was correctly set. Within the Process tab, you then can apply the Auto Detect MEEK version 1 workflow. Keep the default settings and start the algorithm. Like with the disk image R analysis, you can open the results by clicking the Results button at the top right. And again, you can download and browse through the results. This concludes the tutorial for the J-AST platform. More documentation can be found online and directly within the software's help panel.